I'd like to welcome everyone to today's edition of the Polyploidy Webinar. Today we have two speakers, and up first is Dr. Verónica Di Stilio. Verónica completed her undergraduate degrees in Buenos Aires, and she earned her PhD in plant biology at the University of Massachusetts, working on plant sex chromosome evolution with David McCauley. Her first postdoc was on pollen gene expression and the role of Superman on cell division with Alice Chung at UMass, and a second with David Baum and Elena Kramer at Harvard University, where she was introduced to the field of evolution of development. She joined the faculty of the Department of Biology at the University of Washington in 2003, where she is currently an associate professor. She continues to pursue her interest in the evolution of plant development with emphasis on flowers. And today she's going to tell us about her research at this wonderful intersection of polyploidy and reproductive systems, and tell us about the interdependence of polyploidy, unisexual flowers, and wind pollination in a ranunculid. With that, I'll let Verónica take it away.
Thank you, Mike, and thanks for the invitation. Thanks everyone for being here. I will talk about interdependence of polyploidy, unisexual flowers, and wind pollination in a ranunculid. And in this first title slide, I should say first that this is a collaboration with Rosanna Zenil-Ferguson, who is at the University of Hawaii, and also with a graduate student, Simra Sahib, from here, the University of Washington. So in this first title slide, I think you can start to appreciate that this plant that I'm going to tell you about has quite a variation in ploidy levels. So you can see diploids with 14 chromosomes here at the bottom, all the way up to more than 200 chromosomes towards the top. And also that seems to be correlated with changes in the mode in which they're pollinated, from insect pollinated flowers like this one here to wind pollinated flowers that tend to have separate sexes, like this staminate flower and carpellate flowers here. And some examples of what they look like when they're insect or wind pollinated on the right. So my lab has been interested always in understanding developmental genetics of plant innovations. I've been focusing on the flower as the key innovation for the huge radiation of the angiosperms. In particular, I've been focusing on ABC model for the last decade or so, and also some other flowering genes that are above in the cascade. But for today, I'm going to tell you about polyploidy in this system, which is a eudicot, which is where most flowering plants are. You could think of it as a mini radiation within the angiosperms. And it's in the order Ranunculales, which is sister to all the other eudicots. Within the Ranunculales, there's the family Ranunculaceae and the genus Thalictrum. And these are basically the three main types of flowers that can be found in the genus. They don't have petals, and so they either have petaloid stamens, petaloid sepals, or they're small and green and inconspicuous in the wind pollinated ones. And this one here is the one where we've been doing all of our functional studies. It has also been tackled by 1KP. And we've done so because it's diploid and it has a small genome size. And also we've been doing targeted gene silencing in it. So it's been our model of choice within the genus. We've been studying also duplications in the floral organ identity genes. Those are not related necessarily to the whole genome duplications I'm going to tell you about today. The genus has about 200 species, and it has this amazing ploidy series from 2x all the way to like 12x that goes along, like I said, with this diversity in shifts to wind pollination and unisexual system. So here again, we're showing you some of that diversity. The insect pollinated flowers on the top are always hermaphrodites, and the wind pollinated flowers can be hermaphrodites like this one here with its stamens, carpels, and sepals, but much more reduced and green. And they typically have sexual separate sexes, like in this dioecious one with staminate and carpellate plants, or this andromonaceous one with hermaphroditic and staminate flowers on the same plant. And then they have this amazing series of ploidy from diploids to from 14 chromosomes to more than 200 chromosomes. So we've done several reconstructions of these characters in the past. It's always been the main thing that we've been focusing on is this amazing transition from the insect pollinated to the wind pollinated types of flowers, which you can see are very different in the way they look and they conduct themselves. And with the likelihood reconstructions, we have shown that wind pollination has evolved repeatedly in this genus. In this phylogeny already, we capture these main two clades, and clade 1 has all the, what we think is all the ancestral features for the genus, which is being diploid, insect pollinated, and hermaphrodite. And it's in this clade 2 that polyploidy explodes, and so do many other transitions, including to wind pollination and to unisexual systems. So other reconstructions that we've done in the past for Ploidy, for example, have also shown that this clade 1 is solidly diploid. And then there's 3 main clades that at the time we weren't distinguishing. We didn't have a very good resolution, but now we do. But these always hold, and one is mostly diploid, the other one's tetraploid, and the other one has polyploidy going ballistics in it from a haploid number of 7 all the way to 105. And so we've seen that polyploidy has occurred frequently. Relatively recently, the genus has 18 million years, and this clade 2 where polyploidy comes in is about 15 million years. And also when we look along these branches, they're usually associated, polyploidy here is associated with wind pollination and andromoniaceae or dioiceae, which are the unisexual sexual systems. So we were observing early on this connection, and hence we wanted to look into that a little more. And of course, there's this diploid polyploid and this correlation between wind pollination and polyploidy that we observed statistically early on. So now we have a better phylogeny. We have about 50% taxon coverage. And even though it's only 6 chloroplast loci, they've been very informative, and they're able to resolve that backbone of the phylogeny that needed better resolution. So with this new phylogeny and considering lineage diversification as part of the process, we want to reassess the questions that we had about how polyploidy evolved in this genus and presumably how it interacted with the evolution of wind pollination and unisexual systems. And there are some quintessential questions that we can tackle with this, like does polyploidy promote diversification? Typically, one hears that insect pollination promotes speciation, but is that the case in this system where plants have, lineages have transitioned to wind pollination repeatedly? Also, there's been transition to unisexual systems. So is it a problem here, or is it more like an advantage? And we also want to get to the order of character evolution. For example, are transition to unisexual flowers more likely in a wind pollinated background, or did unisexual flowers evolve first? So those are some of the questions that we want to reevaluate under models that consider lineage diversification and trait integration. So that's the key, that we want to integrate these traits, and we want to look at them in the context of how the lineages are diversifying. And for that, and with the help of Rosanna, we've been using these state-dependent speciation and extinction, or SSE models. And in their simplest form, this model takes a focal trait, for example, in this case, ploidy, and considers its 2 states, diploid, polyploid, the transition rate between them, which is rho, and we consider it non-reversible. So diploidization exists, but we don't consider it's something that's happening in our system. And then there's a extinction rate and a speciation rate. Okay, and in the same way, we have the 2 other traits with their character states. Some are more reversible, like pollination. Transitions to wind can go back and then go to insect. The HESI hidden state model will take into consideration noise. So it's a much more realistic analysis, which considers that it's not just the focal trait that is affecting diversification, but potentially other background noise is there as well. So more realistic scenario. So now we have the same two states, but we have in A or B, or hidden state B. And then when we start to consider them all together is when we get more realistic, and that's the MUSI or multiple state SSE. And so, for example, for pollination and ploidy, we now have 4 possible combinations. You can be insect diploid, insect polyploid, wind diploid, or wind polyploid. And then that same combination can be looked at with the noise or hidden state, and that's the mu-HESI. So you can see how it becomes quite complicated fairly quickly. So let me show you some results of these analyses, and we've done all of these combinations. The next step will, of course, be to do the 3 together, but so far we've done the combinations of 2 traits. I also wanted to point out that we customize, of course, the model to the biological system. And so, for example, there's one here that doesn't exist. So we don't have the diploids that are unisexual. All the unisexual taxa are polyploids. So when we run the simplest scenario, which is the binary state, just 1 trait at a time, we already see some interesting signal where polyploid taxa that are unisexual and wind pollinated are diversifying at higher rates. So these are diversification curves, and we're looking at them relatively speaking, relative to one another. And we're seeing that it's these types of lineages that are diversifying at higher rates. When we consider the hidden state, that's still the case. So that's interesting that even when considering noise and background in the system, both polyploidy and unisexual systems and wind pollination are still diversifying at higher rates. When we start to combine them in the multiple SSE, I'm going to show you some examples. So for example, ploidy and sexual systems. So now we have polyploid unisexuals here in the aqua are diversifying at higher rates. And that is still the case after we consider a hidden state. We still get both in state A and B, the polyploids that are unisexual diversifying at higher rates. So unisexual polyploids are winning here. When we look at pollination and sexual system, we can see that there are differences between insect and wind pollinated taxa, but not so much by sex. So we have here the 2 wind and the 2 insects. So the wind pollinated lineages are diversifying more. But interestingly, when we look at the transition rates, we can see that it's more likely to transition to unisexual systems in a wind background, the orange here, than it is in an insect background. And that's interesting because it might already give us a hint about order. And back in 2012, we had this hypothesis of, okay, how do you go from a hermaphrodite insect to a wind pollinated dioecious? And you could go by evolving first through wind pollination or through dioecy. We already thought that this was a more likely scenario for our system, and this seems to support that. But let me show you a little more of other lines of evidence that might support this hypothesis. First, I want to tell you about pollination and ploidy. So here also, the wind pollinated group are diversifying at relatively faster rates, but they're also showing background noise. So there's other things going on. And when we look at these in the ancestral state reconstructions, which is what the analysis is doing, it's reconstructing on the tree, that gives us some hints about order. So for example, it tells us that the ancestral state for the genus, these are the outgroups, is insect and hermaphrodite. And then there's a transition to wind pollination in the green before going to unisexual systems in the yellow. So that already suggests that perhaps wind pollination came before unisexual systems. when we look at, and that again is consistent with this hypothesis highlighted here, hypothesis 1. When we look at ploidy and sexual system, the ancestral state is reconstructed as diploid and hermaphrodite. So luckily, all the ancestral states are reconstructed the same as we had before with likelihood. And we see that there's a transition through orange to aqua. So the orange means that they're becoming polyploid before they're becoming unisexual. So we have a transition to polyploidy before unisexual system here and here. Not in this clade, and that could be that we're missing in our sampling some polyploid hermaphrodites. We'll have to see if that's the case when we increase our sampling. But in any case, it's looking like polyploidy came before unisexual systems. So putting that together at this point, if you're asking yourself, well, what about polyploidy and wind pollination? They're happening very close together for us to tell. And I'm showing it smaller here because we can't quite tell. But still, the ancestor reconstructs as insect and diploid. And then we get these transitions to wind-pollinated polyploids pretty much solidly here and here, except for two that go in the two directions, these two little clades here. So we can't tell for sure at this point. It looks polyploid and wind pollinated, our pollination are pretty tight together. But we have some ideas from the biological perspective of why perhaps polyploidy first makes a little more sense. It's possible that polyploidy allowed these plants to be more resilient at times when there were extreme changes in global weather, and also to become taller, and that may have had an advantage in subsequent evolution of wind pollination. In fact, the diploids tend to be under a foot tall, whereas many of the polyploids can be 5 to 6 feet tall. In conclusion, the ploidy level and sexual system are very relevant in Thalictrum diversification. Wind-pollinated unisexual polyploids are diversifying faster than the insect-pollinated hermaphrodite diploids. Luckily, all the ancestral state reconstructions are consistent from the early on when we were doing likelihood to now when we're integrating everything. And it reconstructs the ancestor for Thalictrum as diploid, insect-pollinated, and hermaphrodite. And we are thinking that we're leaning towards polyploidy probably came first, but we can't exactly tell with wind pollination occurring very closely, and then followed by unisexual flowers. So with that, I would like to thank the NSF-DEB Opus Mid-Career Synthesis grant that allowed me to collaborate with Rosanna and learn these analyses that have given me a completely different macroevolutionary look at a system where I had been doing more microevolution until now. And student Simra Sahid has been instrumental in running this. And then former people in the lab that I've shown data from, Valerie Sosa, who was a postdoc, and Jesus, who was an undergrad and is now a grad at Cornell. Thank you.
Well, thank you, Veronica. That was wonderful. As usual, folks, if you have any questions, please put those in the chat box and we'll take them in the order, and you can turn on your camera and mic and always ask them. It's a little nicer to actually hear and see people.
Yeah, I have the chat up here so I can. Oh, hi, Madeline. Thank you.
Hi. I have a question. Can I just ask my question? Sorry.
Go for it. That's perfect.
Okay. I have a question. So this repeated evolution of wind pollination, what do the flowers look like? Do they always look the same? What does morphology look like when wind pollination evolves?
Yeah, that's interesting. We're also very interested, and we're trying to, we're doing PCA analysis of floral traits to tackle that. And yes, they tend to converge. Let me see if I have it here, actually. Okay, so Yeah, so we're doing this PCA analysis, and we do see traits that converge into things that look like this alpinum here. So the small sepals, the longer filaments that are skinny, and the bigger anthers. But there are some intermediate features too. So this one that is shown here kind of looks like the wind ones, but it has some colors. So there are some that actually in the PCA are looking intermediate, and there's some evidence that there's some ambophyly, so there's that too. But yeah, we'd like to figure out the genes, so we're trying to get to the traits that are most important, so then we can find some candidate genes as well.
Great, thank you.
Oh, sorry, let's see. So Carl is asking, or should...
Either way, I had my mic off. I actually told Carl to go ahead and talk, and then realized I was muted.
Okay.
So if Carl wants to unmute and go ahead and ask.
Okay.
That was super cool. Thank you very much.
Sure, thanks.
A very basic question. The models let you look at the effects of the traits individually and in combination, but they don't let you look at the effects of a trait controlling for the effects of the other traits. That wasn't phrased as a question, but that was a question.
Okay. I'm not sure if I understand. So we are looking at in combinations of two, and we will be looking at the three together as well. You're saying if you can tell by looking at any one if the others are having an effect.
Let me rephrase, sorry. So you can demonstrate an effect of being wind pollinated and being polyploid together. Okay. But can you determine whether there's any effect of, say, polyploidy individually, controlling for the effect of pollination syndrome, pollination?
Yeah, I'm not sure. I guess when we're looking at the individual ones, in a way, we're doing that. But is Rosanna here maybe to help me out?
Yeah, I'm sorry. There's a lot of noise in my house. But I can talk about a little bit about that. So
thank you.
If you wanted to see, for example, the effect of two by controlling the third one, there's not like official stats that can do that, but there's model comparison analysis that will allow you to measure the contribution of one trait to the diversification process. And that will be done by writing a model that contains the three of them, and then the reduced model that is the one that contains two of them, and comparing those two. And if it turns out to be significant, the one with the three traits compared to the one to the two traits, then you can speak about the contributions of the third trait. But there's no official stats to control right now, or official models that can control or fix, right? Or fix even the direction of things. So yeah, these models are really still on diapers, I would say. They are just starting to pick up to be more complex and represent more biological histories.
Thank you very much. In a case like this, where the traits are highly correlated empirically, it'll be very hard to say anything.
Yes, and you see that in some other systems where, for example, you have a change in breeding system along with the polyploidy, and they are completely coupled, right? In which case you cannot tell whether it was polyploidy or breeding system first. But in Thalictrum, it doesn't seem to be the case. There's still some uncoupling there that in which you can see that there's some combinations that are better than others.
Thank you. There's another question by Paul.
Yes, Paul Allen. Go ahead, Paul, if you want to ask your question.
Yes.
I apologize first because I'm not a botanist and my question may reveal a certain degree of ignorance. And it's not about polyploidy, but how solid is the evidence that insect pollination is the ancestral state? It seems like the development of specific insect attractant structures is a much more complex system than having a random wind vector for pollen. Could you address that?
Yes, in our system, it seems pretty solid that insect is the ancestral state. Insect pollination is also present in the outgroups, so other ranunculaceae. And one thing that relates maybe to your question is that Thalictrum may be transitioning so easily to wind pollination because in a way it has some acceptations, this idea that it already doesn't have petals, it doesn't have nectar. So it's kind of ill-equipped for insect pollination to begin with. So I think it's almost like a path of least resistance in this case. The reconstructions seem to all coincide that the ancestor of Thalictrum was insect pollinated. So that is quite solid in the models. And the reason I think it's transitioning so easily has to do with what they look like to begin with.
So your evidence is based on genetic reconstruction?
It's based on these, well, we did first likelihood, so ancestral character state reconstructions, like simply one by one, and then combined with these models I just described. So yeah, it's based on the phylogenetic comparative methods at this point, using the signal in the phylogeny together with the character states.
Thank you very much.
Yeah, you're
That
welcome.
helps.
I'm going to jump in before Carl's next question real quick with my own.
Okay.
So what happens if you make, what happens if you make a synthetic polyploid in one of these species? Do you see a switch in mating systems, or is there evidence of populations of some of these species that have, different cytotypes and or you said that earlier, but also, is there evidence then that the unisexual, incipient unisexual flowers and some of the populations that are also hermaphrodites? Do you have that kind of variation to work with, and what do you see if you do?
Yeah, I haven't done artificial polyploids, but I mean, this also relates a little bit with the question of sex determination. In the past, Westergaard had done in the 1950s some crosses to try to figure out the sex termination using also like polyploids and diploids. So it is likely that there's a connection there, but I think to study that we could use some species that have cytotypes. That would be awesome, because there are changes sometimes within a species where you go from diploids to polyploids, and I think that would be an opportunity to see if there could be some like zone where you start to see changes in the sexual system going along with the ploidy. But I haven't done it artificially. I think it would be good to use those natural systems to look into it. But yeah, there could be something there. So the sex determination system related to that, since Carl asks, I started working on Thalictrum dioicum because I was working on sex chromosomes, and I thought there could be a close connection between homeotic sex determination, because they are unisexual from inception instead of selectively aborting. So that's why I started working on dioicum. But I have to say, it's been very elusive. So they don't have heteromorphic sex chromosomes, but they do have 1-to-1 sex ratios. So it does look like there's a strong genetic component. It's just not visible at a cytogenetic level. So it has to be pursued these days, I think, with sequencing. And there are people that are working, a student of David Baum is actually working on that at this point in dioicum, trying to find the genetic elements. So there's a genetics system, but it's not heteromorphic sex chromosomes.
Very cool. Well, thank you, Veronica. I think that we'll wrap up there for her. And if there are any other questions or anything else pops in anybody's head in the meantime, we'll have some time at the very end of both talks to ask those questions.
Okay, so I'll stop the share.
Yep, and let Juan take over. All right. And with that, I'd like to introduce our second speaker today. This is Dr. Juan Losada. He graduated in biology and food and science technology at Vigo University in Spain, and did his PhD work at the University of Zaragoza, focused on the reproductive biology of apples. He was a postdoctoral fellow in the laboratory of Doctor of Ned Friedman at the Arnold Arboretum at Harvard University, where he worked on the evolution and development of angiosperm seeds of the ANA grade. He then switched to a research fellowship position located both at Harvard and Brown universities. And at Brown, he worked with Dr. Andrew Leslie, exploring the development and evolutionary trajectories of conifer seed cones, as well as the laboratory of Dr. Missy Holbrook at Harvard, where he developed a strong interest in photosimulation transport toward reproductive structures from an organismal perspective. He then returned to the south of Spain 2 years ago as an independent researcher in the as a ComFuturo in the Spanish Superior Research Council, where he started to build his own research group. And his work seeks to understand the physiology, anatomy, and reproductive mechanisms of subtropical fruit trees with different ploidal levels. And with that, I'm going to let Juan take over and tell us about this really interesting work that he's been doing from individual cells to organisms, source to sink transport in subtropical fruit trees with different ploidies. Thank you, Juan, for speaking today.
Thank you very much. Thank you for your kind invitation, and also thank you all for being here. Today, I'm very happy to talk about our work on subtropical fruit trees and ploidies, and I'd like to start by the motivation of my work. So one of the major challenges, wait, can you hear me? There is something wrong.
We can hear you.
Okay, but I can't. Okay, so one of the major challenges that we face as humanity is trying to combat the harmful effects of the climate change. For example, the lack of water available for irrigation. And perennial fruit crops are excellent systems to test these effects of climate change, because they constitute 1/8 of the total cultivated land. And actually, we can explore the different genotypes available. In particular, subtropical fruit trees within this huge biodiversity, it's kind of unexplored. For example, in the Asian and American continents, in the tropical areas, there are so many subtropical fruits that are locally consumed, but actually we don't have them in the temperate areas of the world. So just to start from the basics, I'm interested in knowing in detail the vascular system of woody organisms in particular. And just everyone knows that there are two major tissues in the vascular system, the xylem and the phloem. The xylem transports the water from the soil to the atmosphere. And in parallel to the xylem, there is the phloem tissue, which transports the photoassimilates from the photosynthetic organs to the growing areas of the plant. And actually, both of them work as individual tubes, single tubes all along the plant. But the interesting part of them is in the individual cells composing them, which are basically microscopic. So when we look at how much we know about the xylem and the phloem, there is a total imbalance of the knowledge, and this has some historical reasons. So we know the xylem for centuries. Mel Piggy already talked about the tracheids in the leaves of some plants and comparing them with the tracheids of insects, but we didn't know much about the phloem until the 20th century, when Ernst Munch developed the theory of how the phloem works. This yin-yang kind of thing in woody organisms is also at the structural level, because the xylem is composed of dead cells with rigid walls, whereas the phloem is composed of living cells with flexible walls. But also at the functional level, the xylem is working under negative pressure, whereas the phloem is working under positive pressure. So the hypothesis of Ernst Munch, the German botanist, is the osmotically generated pressure flow, which is based on the idea that the phloem is a continuous tube composed of individual cells connected in series. And so in the source organs, the photoassimilates will accumulate within the cells of the phloem, creating an osmotic pressure, and thanks to the entrance of water. And at the same time, in the sink tissues, there is an exit of the osmolytes and the exit of water, generate a drop in the pressure. The differential pressure will generate a flow, and this flow has a velocity. And the simplest explanation of how this velocity works is the Hagen-Poiseuille equation, which states that the velocity is directly proportional to the differential pressure between the sources and the sinks, inversely related to the viscosity of the liquid within the tubes, the length of the tube, and directly related to the conductivity of the tubes themselves. And this is what I'm going to talk about today, the conductivity of the tubes, because it's a parameter that it's related directly to the geometry. And so we know that the phloem in particular changes, the individual cells composing the phloem change their geometry in the trunks of different trees composing the temperate forests. And this is actually very interesting because this allows the trees to grow in height. And so we know that the individual cell geometry is key for the functionality of the vascular tissues, particularly the phloem, but we don't know what is correlated with the geometry of the cells. Actually, there is not a lot of information about the phloem in the leaves, and we are advancing a lot on understanding how the different venation, how the phloem scales in the different vein orders of the leaves. And if this is not really well understood, even less information is available on the phloem in the reproductive structures, and nothing really comparing different plants with different ploidies. So my work is based on two different systems. One of them is an auto-tetraploid, and the other one is an allopolyploid. One of them is mango, and the other is sugar apple. Both of them have different evolutionary histories. And basically, our major goal is to model source-to-sink transport in these woody systems with the purpose of understanding whether there is an effect of ploidy in tolerating drought. So we are based on the south of Spain, and there is a very narrow area where we have subtropical climate. And actually, this is very important because there are crops, subtropical crops that are very relevant for this small community. And in our institute, we have a very interesting germplasm collection, including some avocados, mangoes, or cherimolas, and we use this material. So basically, we used Mangifera indica and Annona cherimola. We have 2 ploidies for mangoes and 3 for Annona cherimola, and I'll get into details of each of one. So in mango, we have, we say it's an autopolyploid because we used molecular markers combined with flow cytometry, and the variety we use is Kensington, one of the few varieties that is available with a polyploid, with a tetraploid in this case. And so we started to compare, and we wanted to understand what is the relationship between the physiology and the anatomy of both diploid and tetraploid mangoes of the cultivar Kensington. From the physiology perspective, we started to evaluate the water potential of the leaves, and in the months we evaluated that, we couldn't find any differences in the water status of these leaves. In contrast, we actually found differences in the stomatal conductance, and actually the diploids had higher stomatal conductances in the hours where the weather was hottest. And this was actually correlated with a smaller guard cell length of the diploids compared to the tetraploids. Actually, the transpiration was higher in the tetraploids compared to the, in the diploids compared to the tetraploids, but this was not the same for the water use efficiency. The tetraploids apparently use the water more efficiently than the diploids. So how can we relate these physiological parameters with the anatomical parameters of the phloem? We started to divide the plant into the different functional areas, the inflorescences, the branches, and the leaves. So this will be a cross-section of the mango stem, and we evaluate the individual sheath developments, analyzing the different parameters of length, radius, sheath plate number, sheath plate size, or the pore sizes. And our first observations were that the individual flowers of the tetraploids were significantly larger than the flowers of the diploids. And actually, when we followed the development of the inflorescences from the hundreds of flowers that each inflorescence has, only a few set fruit. And normally, those are located in the tips of the inflorescence rachis. That's why we started to sample these 2 positions in the inflorescence. The top, we call them top and bottom. And so when we evaluated the sieve tube elements, the first observation is that the rachis behaves like a stem, and so the sieve tube elements in the top were smaller than the sieve tube elements of the bottom. But also there is a trend that shows that the sieve tube elements are larger in the tetraploids compared to the diploids. So when we do it for the branches, we actually divided the branches into their different thicknesses, and we see a scaling relationship of the sieve tube elements, the geometry of the sieve tube elements with the diameter of the branch. And actually, this scaling relationship is also consistent with the idea that the tetraploids have larger sieve tube elements than the diploids. Then for the leaves, we sampled the petiole, the primary vein, the secondary and tertiary vein orders. And actually, we observed the same scaling relationship of the sieve tube elements when we go from the tertiary order veins to the primary order veins, with the exception of the petiole where the sieve tube elements get shorter for some reason, an observation that we saw in other species as well. So we not only evaluate the geometry of the tubes themselves, but also the connection between them. And this is just to give you an example of how we fish the individual connections between the tubes. So we locate the phloem tissue, which is a complex tissue, and not all the cells in the phloem are tubes. So we need to locate the sieve plates connecting the individual tubes. And we measure the pores, because the pores are very important for the conductivity, since they offer a lot of resistance to flow. So when we evaluate the pore size along the different pathway of the sap, we see this scaling relationship in the pore size. And the different symbols that you see correspond to the different functional areas. So basically, the tertiary order veins have the smallest pore sizes, and the highest pore sizes are actually in the thickest stems. And we didn't find differences between ploidies in the pore size. So with all this information, we are able to model the transport in the sense of how much resistance the whole tube offers to the subflow. And so what we observe is that the diploids have a higher resistance to the subflow compared to the tetraploids. And this is a work that we have in preparation. So as conclusions, we can say that the sieve tube elements of tetraploid mango trees have larger geometries than the diploids. And this makes that the tetraploids have lower resistances to photoassimilate transport than diploids. And this our observations can be correlated with these differential conductivities from source to sink can imply that there are some organs that get larger, for example, the flowers. So this was for the autopolyploid, but we have an allopolyploid, and the allopolyploid is a product of several years of research. So basically, it's a hybrid product of an interspecific cross between Annona cherimola and Annona squamosa. So the F1 hybrid showed a progeny with two different ploidies, diploids and triploids. And by crossing them with the mother plants, delivered a new hybrid, which is the Atemoia, and we have diploid, triploid, and tetraploid individuals. So this is the material we started to play with, using the ability of trees to be grafted. So we wanted to play with ploidy as a factor, but also with water scarcity as a factor. So we first grafted the different varieties with different ploidies as varieties onto diploid rootstocks. And then we set up an experiment in the greenhouse where we have several plants of the different ploidies, and we applied two different treatments. Some of them with abundant water, and some of them with what we call water stress or water scarcity. We control for the humidity of the soil and other parameters in the greenhouse, and we started to follow the same pathway that we used for mangoes. So we started with the physiological parameters, and this is how the plants look like in the greenhouse in 2018 and 2019. We started the experiment in the 18, and they grow quite quickly, actually. So the photosynthetic activity was maximum for the triploid individuals. And actually, there was a clear effect of the water treatment. So basically, as we increase the water offered to the triploids, they increase the assimilation rates. Similarly, the stomatal conductance increased with the water applied to triploids and diploids. But actually, the tetraploids were not responsive to the treatment, which is interesting. On the other hand, the internal CO2 concentration was the highest in the diploids without differences between treatments, but actually the diploids were the least conservative from the perspective of the internal water use efficiency. So putting it simple, basically we have the triploids which have a lot of vigorosity, followed by the diploids and followed by the tetraploids. And so we also measure the anatomical parameters. Basically, we started with the stomatal size, and there is this direct correlation between ploidy and guard cell length, as it's been observed in several species. But also, from the perspective of the phloem, the sieve tube elements were also highest or larger and in the tetraploids and triploids compared to the diploids, with the exception again of the pediole, where it's an area of the leaf where the sieve tube elements get shorter for some reason. When we look at the diameter of the sieve tube elements, we find the same pattern. Basically, the higher ploidies have larger and wider sieve tube elements compared to the diploids. And this is happening in the major veins, but also in the minor veins of the leaves. So this is for the leaves, but actually the flowers of Annona cherimola are solitary, so they are not terminal as we observed in mangoes. And so they are basically axillary flowers, and they share the same bud with the leaves. So basically we wanted to explore the pathway in the area in which all the photoassimilates will go toward the flower, the pedicel. When we observe the sieve tube elements in the pedicel, we actually find this scaling relationship. Again, the diploids have shorter sieve tube elements compared to the triploids and the tetraploids. And finally, we also applied the same system to the stems, but in the stems, we find a different pattern. Basically, the sieve tube element length was not scaling as much with the diameter of the stems, only in the tetraploids. And then the radius of the sieve tube elements did not differ between ploidies, even though they scaled with the diameter of the stem. We also measure the pore size in the different branch diameters or stem diameters, and we couldn't find differences, clear differences between ploidies, but actually we did find the scaling relationship. So as the branches increase in width, the pore sizes are larger. And so we model again from source to sink, but in this case, we didn't take into account the branches because, as I said before, the flowers share the same meristem with the leaves. And so when we model from the minor vein orders toward the pedicels of the flower, we find that the hydraulic resistance of the diploids is higher than the triploids and tetraploids. And this is very linear. I would say there is a linear correlation in the different vein orders of the leaf. But when we reach to the pedicel, to the pediole, sorry, there is a bottleneck. So there is an increase in the resistance to subflow. And actually, this resistance gets attenuated toward the flowers. So the conclusions for Annona cherimola is that triploids and tetraploids have bigger sieve tube elements in leaves and flowers, but not in the stems. And we think that triploid cherimolas are more efficient transporting the photoassimilates. And this is reflected in the morphological characters that we also evaluated in the greenhouses, because triploid cherimolas are more vigorous, transpire more, and are more efficiently assimilating CO2. So we believe that the triploids are potential water savers. but also the fact that tetraploid cherimolas are less vigorous, but unresponsive to water deficit could be very important to make them potential drought-tolerant rootstocks. So this is the results I wanted to show you today, but also we are working and we are very excited in different applications of our system. So we have the triple graft. Basically, in a single diploid rootstock, we grafted the diploid, triploid, and tetraploids. And actually the results are very interesting because each variety, even though they are grafted in the same rootstock, keeps its own morphological and physiological characteristics. So that leads to the question of what happens if we make the other way around. And so we also grafted triploid and tetraploid, so diploid varieties on deep triploid and tetraploid rootstocks. And we are evaluating the physiological parameters of those under different irrigation treatments. So with that, I'd like to thank all the field workers that are involved in this project, which it will be impossible to do that without them, and all the collaborators, the head of the department, Iñaki, my students, I'm sorry, I don't know what's happening with this. My students, Miquel Barceló and Andrés Fonollá, Elsa Martinez Ferri from IFAPA Andalusia, and international collaborators Carl Jensen from Technical University of Denmark and Missy Holbrook from Harvard University. And thank you all for your attention.
Thank you, Juan. That was awesome.
Thank you.
I really enjoyed that. Any questions? As always, put those in the chat box. I think that the sort of the Frankenstein grafted ploidal levels is perfect for the Halloween week that we're entering here. So
I agree with that.
Veronica has the first question there. I was actually wondering the same thing.
Yeah, they're sterile.
Can you say it again, please? I couldn't hear you.
Not because you were going to read it. I was wondering, those 3N that are very vigorous and they look great, but aren't they sterile?
They are.
Okay.
So we actually started to make some interploidy crosses, and they are sterile when they act as a female, as the mother, but not when they are as the father. So triploid polling will be viable, but not as viable as diploids and tetraploids. But when the triploid is the ovule or the female gametophyte, then they are always sterile.
Okay, thank you.
Yeah, Paul asked, they're sterile and without fruit.
So the mango tetraploids are sterile because they never produce fruit. But we know of other varieties that are tetraploids and they produce lots of fruits. And normally mangoes are self-pollinating. So we think that there is some block in the Kensington variety, but it doesn't mean, so the ploidy is not directly, we can say that the higher ploidy is sterile by itself. But we are checking the reproductive system to see whether Kensington is actually having any problem with the pollen tube growth or fertilization.
Excellent. Carl, go ahead and pop on here.
Oh, super cool. The pattern that you see in phloem and mango, for example, of cell sizes being larger in the polyploids, is that true for all cells, or is there something special about phloem?
So we checked also the size of cells in the leaves, and it's not true for all cells. It's true for the guard cells, for the sieve tube elements and the xylem as well, but not for the epidermal cells, for example.
Cool.
Yeah, I was curious. Do you I'm really excited to see the results of those last stuff that you presented there, showing the changes and the root grafting using the diploid roots or the triploid roots. Do you get a sense that the amount of material being moved is I'm trying to think about this in terms of like the volume, I guess, of synthate being of photosynthate being moved around in tetraploids versus diploids. Are we are these sizes actually translating to sort of more volume being moved or per like a higher flow rate, I guess, is what I'm thinking about, or not? And I'm just thinking about, I've worked on like a long time ago in electron microscopy of trying to get those beautiful images you showed, and not rupture all of those like sieve plates and things, right? And not rupture all that tissue. I'm just thinking, is there a physiological sort of limit here as to how much flow you can get and without causing damage or without having cavitation, that kind of stuff? And just think about it from a physiological perspective. Do you see any evidence that the cells are bigger or smaller than you might expect, given the sort of ratios that you see, for example, of tetraploid to diploid guard cell sizes? Do you see the sieve tubes are more or less constrained in that way? I'm kind of sort of thinking out loud here, thinking off the top of my head.
Mm-hmm.
But
no, it's fine. So in terms of how much subflow exists between diploids and polyploids, there are more factors affecting, as I said in the beginning, because the viscosity may change with the different irrigation treatments. For example, we couldn't measure the viscosity. It's kind of a complicated parameter to measure. But in the beginning, intuitively, you can think that bigger sieve tube elements will transport bulk flow of more photoassimilates. But actually, it's not such a simple relationship. So we should account for the viscosity, and we should account for the pore size in the plates. That's why we are still working in all the individual sieve plate connections at all areas of the transport pathway, because they are actually playing the most important part in the transport of photoassimilates. So those are the important parameters, because they also influence the different pressure between the sources and the sinks. And so altogether, they will be very important. But basically, we are starting by the geometry of the individual sieve tube elements, because it's the basic we can do. And then we will try to evaluate the viscosity and other parameters related to the irrigation. Not sure if I answered the question.
Yeah, and I guess the other way, as you were talking to, I was thinking a little bit about a different way to ask what I was trying to ask. What do you think would happen if you were to examine, say, octoploids and make synthetic octoploids? Do you think that these the curves would just keep going in a sort of this linear fashion, or do you think that they would start to plateau as you increase ploidy level, potentially because of just physiological constraints of changing those sieve tube element sizes, for example?
I think there are physiological constraints, because in order to grow taller, for example, there is we need a significant pressure formed in the source organs. So there shouldn't be a direct relationship with the ploidy, and so there must be some limits, but I would love to explore them. And actually, we saw it in Annona cherimola. There is not like a direct relationship in all organs, only in some of them. So it will be very interesting to explore what's happening with the individual parts. Obviously, yeah, we are also thinking on creating new polyploids and explore the direct effect of polyploidization on the cell size of some areas.
I like that. Oh, there's a couple other questions now. Let's see. So Paul Allen asks, let's see, presumption would be that higher resistance to flow might confer less vulnerability to drought stress. And do you presume the opposite?
So, my presumption will be that higher resistance will flow. It depends. It's always depending on the accumulation of photoassimilates in the loading areas of the phloem. So if there is a higher resistance, but there is a higher volume of transporting similar concentration of photoassimilates, then it will be beneficial for the global growth of the plant. We know that when there is drought, there is an increase in the viscosity, and so there is when the drought gets really extreme, the viscosity is so high that there is no possibility of transport. But I will think that lower resistance will be less vulnerable to drought stress. Yeah.
All right, and then there's a follow-up question there from Ben Gerstner. Benjamin, if you want to come on and ask it.
Yeah, sure. I was just curious, in the allopolyploids, how many unique triploid and tetraploid lineages do you have? Or are each of those cytotypes kind of only represented from a single origination event?
So we have several triploids and a few tetraploids. And from those, we made clonal propagation with grafting.
Okay.
But actually, the triploids are more common than tetraploids.
Mm-hmm.
All right. Well, are there any other questions for either Juan or Veronica today? All right. Well, I think that with that, I want to thank both Juan and Veronica for speaking today and giving us two great talks on, there's always ends up being these sort of fun convergent systems. Today it was this interesting interaction between ploidy and reproductive systems that either the products of reproduction or the reproductive systems themselves. And that was really wonderful. I want to thank everyone for showing up as well. Look for these talks to be posted later today up on YouTube, and I'll tweet those out, a link to the video. But otherwise, please join us in a couple of weeks for the next Polyploidy webinar, featuring a couple of talks on the systematic side of polyploidy, and keep your eyes open for those announcements. And I want to thank everyone again for showing up, and again, thank you to Juan and Veronica for two excellent talks this morning.
Thank you very much.
Yeah, thanks for organizing.
Yeah, well, you're welcome. I have a ton of questions. I'm going to probably end up emailing everybody about these things, and but I really appreciate both of those. Thank you.
Sure,
Thank you.
Yeah, thanks for the invitation.